Do it?
No.
Oh,
We're ready. We're ready.
Okay, we're ready. All right, everyone, let's get started. Let's not delay. Very delighted to introduce Gabriel Katul to today's CPEP seminar. Gabby has degrees from the American University of Beirut, Oregon State, University of California, Davis. He's been on the faculty at Duke University on decadal timescales now, where he's the Theodore S. Coyley Professor of Hydrology and Micrometeorology. While there, he's done numerous tours abroad, as well as other universities in the United States, and has received important awards, including the McElwain Award from the American Geophysical Union, John Dalton Medal from the European Geosciences Union, and extra important to this building and this department, the Outstanding Achievements in Biometeorology Award from the American Meteorological Society. So without further ado, Gabby.
Thank you very much for having me here. Well, thanks, Paul, for this introduction. To begin with, this work is done in collaboration with numerous co-authors and collaborators and current and former students. I'm just going to quickly mention them here. My current student, Asad Murad and Mazen Naad, are going to be the primary source for this material, but this work is done in collaboration with numerous others that are listed here, including most of them. So, I think it comes as no surprise to many of us that plants are the Earth's primary solar energy collectors, and their combined mass has been well established to exceed all other forms of life on the planet. And to achieve this impressive feat, plants had to rely on a range of physical, chemical mechanisms that facilitate long-distance transport of water and assimilants, which would be the primary topic for now. And the reason they had to do that is that in a desiccating atmosphere, gas exchange between the leaf and the desiccating atmosphere is still happening by diffusive processes, but this diffusive mechanism is in fact enabled by a whole slew of hydraulic networks that move fluids and assimilants, primarily sugars, with minimal energy expenditure, meaning no pumping. And how they did that and what sort of conclusions we can draw about the functionality of plants and how it might teach us about other systems is what we will be mentioning today. Now, movement as a topic has been around for a long time. It has been studied by famous philosophers, including Aristotle, who stated all motions is caused and can be sensed. So there are two things to be recognized about motion. There is a certain cause that has to be identified, and it has to be sensed. And we further proceeded to observe that once there is motion, that motion will continue infinitely unless it is stopped. If you think these are precursors to Newton's laws of motion, they are. So these were reformulated by Newton in 1666, primarily inspired by Aristotle's observations on motion. But the idea that motion somehow is connected to some optimality principle started by the work of Euclid and later on by Hero of Alexandria, where the concept of a principle of shortest path was first introduced to explain reflection. And that was used for a long time in optics and still is used today in optics. But then studies on diffraction of light popped up, and it was recognized that the principle of least distance or shortest path no longer suffices to explain diffraction and reflection. The two were united, so to speak, by the principle of least time by Pierre de Fermat. And since then, the idea is that there is some sort of an optimality principle hiding behind perhaps deterministic laws kicked in. Those were articulated perhaps by Pierre-Louis Maupertuis. Maupertuis is credited for being, so to speak, the father of the concept of the principle of least action. This principle now is formulated based on the integral of the so-called Lagrangian, which is the kinetic energy minus the potential energy of a certain particle. This is not how exactly Maupertuis framed it, but this is the current interpretation of it. And basically what the action says is that if you minimize this quantity S, which is defined as an integral of this Lagrangian, then you could recover all laws of physics, in fact. So if you're interested in a particle moving in a gravitational field, well, your potential energy is the gravitational potential, and your kinetic energy is 1/2 mv squared. If you're interested in how a charged particle moves in an electric field, the same argument. If you could formulate a potential energy, and if you could formulate a kinetic energy, then you could derive the laws of motion from this type of optimization. And in fact, Maupertuis went even further to say that the laws of movement and of rest deduced from this principle, meaning the principle of least action, being precisely the same as those observed in nature, we can admire the application of it to all phenomena. And in fact, he listed specifically the movement of animals, the vegetative growth of plants, etc. So he even was already envisioning at that time that there may be some sort of an optimality principles behind perhaps the movement of fluids in plants, or the movement of plants themselves. And he goes further to say that, the movement of animals, the vegetative growth of plants, are only its consequence, the consequence of the optimality principle. And the spectacle of the universe becomes so much the grander, so much more beautiful when one knows that a small number of laws, most wisely established, suffice for all movements. So that is kind of the spirit of this talk. It's captured by this quotation from Maupertuis. Now, just as a concluding point on Maupertuis' work, in fact, Maupertuis' ideas led to the establishment of the principle of least action and its solution using the calculus of variations that are credited to Lagrange and Euler. So these are known as the Euler-Lagrange equations. They were, of course, used by Hamilton to solve a whole slew of problems in physics. And now the question that we can ask ourselves is that, is there something similar that is occurring in plants? Is really Maupertuis' vision about perhaps a general optimality principle, can it be derived for plants? Does it follow some sort of a minimum expenditure or some other optimality principle? Now, to do that, we need to revisit back the movement of fluids in biology, and that also has a very rich history. It started perhaps by William Harvey, who was known for the study of basically blood circulation in mammals, but then he also observed that plants do not have a heart, yet they are able to circulate fluids, and it was not very clear how they could do that. So clearly, William Harvey's view, who by the way was a doctor at Padua University, was clearly aware that somehow fluids are being moved without a heart, and that was a puzzling observation even then. The first attempt to study it actually came from Stephen Hales. This is the famous Hales comet, St. Hale, who also met Newton, by the way, and Newton was the person who commissioned his book that is called Vegetable Statics. He observed that plants imbibe moisture so vigorously up their fine capillary vessels, thereby gives the sap vessels liberty to attract fresh supplies, of course, of water from the soil. Sap vessels are adapted by their exceedingly fineness to raise the sap to great heights. I should say that even though Hale was the first to observe the fact that, yes, capillary forces are responsible for moving the sap, he did not identify the cause of why moisture would be moving up. Okay? He did not clearly identify that. And now we have a much richer view of the problem. So our contemporary view looks something like that. We envision that for plants to accumulate this huge biomass that is occupying a lot of the weight in living matter in the planet, they had to uptake CO2 molecules and produce sugars. And in the process of uptaking CO2 molecules, they had to lose water molecules. This process is not very efficient because the photosynthetic machinery of plants developed actually in aquatic environments, not in a desiccating atmosphere. And what has happened is that as for every water molecule that is lost, in principle, the entire column of water has to be pulled up by one molecule. And that is how tension gets created in the xylem vessels of the plant. So this is basically the delivery of water from the soil all the way up to the leaf. Now, this process of uptaking CO2 molecules is quite inefficient for water, surprisingly. For every CO2 molecule that the plants take, it loses about hundreds of molecules of water. Okay? So it's a very inefficient process. But the upshot of it is that for every loss of these water molecules, you have to replenish it from the soil, and that creates a pull in the xylem. It creates tension in the xylem of the Now, water is being transported through sapwood in the xylem under tension, and because water is under tension, it's in a metastable state, meaning that it is prone to cavitation. And that's basically what happens, and in fact, cavitation also creates loss in conductivity, which will be studied in a little bit later. Now, the products of photosynthesis are also translocated down through the phloem, but unlike the case of the xylem, where water is moving under tension, in the case of the phloem, water is moving under positive pressure by osmosis. Basically, when sugars get loaded into the phloem, This is a membrane, so it doesn't allow sugars to leave, but it allows water to enter. As water enters by osmosis, it pressurizes the fluid here, builds up the pressure that is able to push water and sugars all the way down. So the argument in this talk is that each of those systems has an optimality principle behind it. And one of the holy grails in plant physiology is to come up with a holistic optimality principle that is able to recover all these other processes in these three compartments. So let's make a little bit of a review of what has been done in these optimality principles so far. At the leaf scale, the idea is that stomatal conductance variations may be viewed as a compromise between the need to provide passage for assimilation and the prevention of excessive transpiration. This was put forward by Cowan and Troughton in 1971. These ideas are not new, they were around for a while, and so this is kind of an engineer's view of the problem. It's a systems optimization problem. You have a control variable, that is the stomates. Adjusting the stomatal aperture allows you to update CO2 while minimizing somehow loss of water to prevent excessive transpiration that can lead to failure in the xylem. In the case of the xylem itself, that is now the main delivery mechanism of water from the soil to the leaf, it was argued by Tyree and Sperry in 1989 that plants evolved a variety of strategies to prevent hydraulic failure in their conductive tissues while ensuring efficient water delivery to leaves. So again, there is some sort of a trade-off between efficiency and safety. This hypothesis frames the so-called safety-efficiency trade-off, sometimes called the runaway embolism versus hydraulic sufficiency. So that's another strategy, and plants have many, many strategies that somehow play around with this safety-efficiency trade-off interface. Last, in the phloem, it is argued that phloem vasculature must follow, basically, must be optimized to allow efficient sugar transport. And while this statement looks like only efficient sugar transport, it has to be remembered that increasing sugar concentration to enhance the osmotic forces that can drive water down, you are also increasing the viscosity of the fluid, and that's increasing the resistive forces to that movement. So again, there is some sort of an optimality there because there is a trade-off between increasing the driving force and increasing the frictional force. So what I'm going to do next is just walk through the key elements of these types of optimality principles and then try to stitch them all together. So on the leaf equations, we well understand now that a lot of the complex processes that are associated with the Kelvin cycle can be represented in a very primitive way, at least mathematically using the Farquhar photosynthesis model. The Farquhar photosynthesis model, you could check John Norman's book about that. It's a very good chapter, by the way. Relates the photosynthetic rate Fc to the internal CO2 concentration. And that relation is in fact a saturation function. You could see that you have Ci in the numerator, you have Ci in the denominator. So as Ci increases, Fc saturates. And this is often viewed as the biochemical demand. Whether this biochemical demand is driven by, say, photosynthetically active radiation or temperature, or more formally by Rubisco limitations or RUBP regeneration, depends on the values of alpha-1 and alpha-2. Those vary with light and temperature. So this is one equation with two unknowns. It simply establishes a link between a flux and the store, if you will. It's very common in hydrology. If you work ever with reservoirs, there is always a relation between a storage and an outflow, well, something like that. Now this demand is supplied by the atmosphere. CO2 molecules in the atmosphere will hit the leaf surface, and if there is enough gradient between the internal CO2 and the atmospheric CO2, some of those CO2 molecules will enter the substomatic cavity and then go to the mesophyll cells and then of course participate in assimilation. And so this equation then might be summarized by a Fickian diffusion process or some sort of an integrated version of it, where the flux is some stomatal conductance multiplied by the imbalance between the atmospheric CO2 concentration and the internal CO2 concentration. So by equating the demand to the supply, we do have now two equations and three unknowns, basically. The stomatal conductance is an unknown, the internal CO2 concentration is an unknown, and the photosynthetic rate is an unknown. So clearly we need one more equation to be able to mathematically solve this problem. Yeah? In turbulence, we call this a closure problem, and therefore we need a closure model. Yeah? Since we have an atmospheric scientist this morning. Okay, so two equations and three unknowns. So even if you know the atmospheric CO2 concentration, even if you know the physiological properties of the plant, alpha-1 and alpha-2, you still need an extra equation.
And historically, that equation came from empirical observations. So, for example, Paul Jarvis and many others have proposed models that try to link the environmental stimulus to the opening and closure of stomates. Alternatively, one might argue that perhaps Maupertuis's vision might come into play. We might view potential energy as somehow the internal energy that is needed or stored and is required to be drawn upon to activate, so to speak, stomata so that you could provide energy for photosynthesis, whereas the kinetic energy, well, work must be performed to lift water up, and so stomatal regulations actually may have some connection to kinetic energy. So again, we might envision a Lagrangian that is somehow connected to the photosynthetic rate and perhaps the ability of plants to lift water up from the soil to the leaf. So at some level, even with Maupertuis's view, we might envision that there is some connection between an imbalance between kinetic energy and potential energy, if we can get the units right. But more formally, this has been done, as mentioned earlier, by Cowan and Troughton in a much more elegant way. They said that, okay, for the plant to survive, it needs to maximize its carbon over a certain duration. Didn't specify too much the duration. It didn't matter too much for their optimality also. Suppose the duration that we're interested in is T, and T could be anything. It could be the interstone period, between rainfall events. It could be an average value of that. You name it, could be a growing season. It was not clear in the analysis, or it didn't matter too much, as we shall see in a minute why. But the objective function is somehow to maximize the photosynthetic rate over this period of time. But this maximization is not happening without constraints. There are certain constraints. And the constraint that they have analyzed is the fact that there has to be enough water in the rooting zone to support this photosynthesis. So therefore, the integral of the water vapor flux, or the transpiration rate from the leaf, has to be balanced by the stored water in the rooting zone. I should be careful here. It should be the water stored in the rooting zone per unit leaf area, because this is a leaf level equation. And that's a constraint optimization. It's not easy to solve. But thanks to the work of Hamilton and Lagrange, we have a way to solve this constraint optimization problem. And the trick is to convert them to an unconstrained optimization problem. And the way you do that is you introduce a Hamiltonian, which is based on the objective function that depends on the control variable. And again, our control variable is the stomates, the stomatal aperture, if you will. And the constraints suddenly become adjusted, by a Lagrange multiplier that in ecology or in plant physiology, it is called the marginal water use efficiency. And so you could write a Hamiltonian, basically, very much like Euler and Lagrange did. And in fact, you could even write the Euler-Lagrange equations themselves on this Hamiltonian. Now, fortunately for plant physiology, is that the Euler-Lagrange equations are complicated, and they are a differential equation because they do depend on the rate of change of the Hamiltonian with respect to the rate of change of the stomatal conductance with time. However, all the control variables that we have looked at do not suggest that the rate of change of stomatal conductance itself with time is a factor. So in essence, this formulation suggests that the photosynthesis is only a function of stomatal conductance. The water vapor flux is only a function of the stomatal conductance and not the rate of change of the stomatal conductance with respect to time. So under those conditions, the Hamiltonian is not dependent on the rate of change of stomatal conductance with respect to time, and this term disappears, leaving us with the rate of change of the Hamiltonian with respect to the stomatal conductance to be zero. So under those conditions, we can formulate this as a hypothesis, as a closure model to the original system, which is an optimality principle. So with that, now we revisit the same problem one more time. We have a biochemical demand that links photosynthesis to internal CO2 concentration. We have an atmospheric supply that links photosynthesis to internal CO2 concentration. But that's a fake additional equation because it's introducing one more unknown, the stomatal conductance. Well, now we have an optimality hypothesis that says the rate of change of the Hamiltonian with respect to the control variable stomatal conductance has to be zero. In this analysis, the marginal water use efficiency, or the Lagrange multiplier, that is really converting the water economy to the carbon economy so that you could operate in the same units. The temporal changes of those are momentarily neglected. I will mention briefly how they can be brought down. And in principle, we should be able to solve for the Lagrange multiplier, lambda, through the constraint. I mean, this is how it was derived in the first place. So you could integrate the water flux over a certain time period and set it to the maximum water that is available in the rooting zone per unit leaf area. And that is basically the constraint. And the problem in principle is mathematically closed for the equation and for the unknown parameter number. In practice, we really don't know what the integration period is. We really don't have a good sense of what the initial soil moisture content is, and we really don't know whether this constraint is fully valid. So if you assume the marginal water use efficiency to be constant, you are basically ignoring this constraint here and simply treating the marginal water use efficiency as an empirical factor, which is what has been done in the early work of Kahneman and Feynman. Now, to give you a sense of what these solutions look like, I've picked a linear biochemical demand function, meaning the photosynthesis is linearly related to the internal CO2 concentration, and this linearization is simply removing the internal CO2 concentration from here and replacing it by some fraction of the atmospheric CO2 concentration. So if you combine this linear biochemical demand function with the Fickian diffusion and the Hamiltonian principle that is to be maximized, you could predict the stomatal conductance. And there are several interesting features about this prediction. The first is that it shows that stomatal conductance does vary universally with vapor pressure deficit, with a scaling exponent that is 1/2, or minus 1/2. It also predicts a relation between photosynthesis and atmospheric CO2 concentration and vapor pressure deficit. And no less interesting, it predicts the internal CO2 concentration to the ambient CO2 concentration to be some dependent variable on vapor pressure deficit. Notice all of these power laws are emergent properties of the optimization. They were not a priori assumed. And with a little bit of work, you could see that if you divide photosynthesis by GS, or you take the slope alpha 1 divided by alpha 2 plus SCA of this quantity here, and you rework it back in here in terms of photosynthetic rate and a little bit of algebra, gives you a relation between stomatal conductance, photosynthesis, and atmospheric CO2, an aridity index d to the minus 1/2 that is completely predicted from optimality principles, and the multiplier here that depends on atmospheric CO2 and the Lagrange multiplier. Now, this was derived for a linear biochemical demand function. Melinda Bedlin also proposed a similar mechanism for RUBP regeneration, where the photosynthesis internal CO2 concentration is a little bit more nonlinear. But both approaches lead to a similar universal pattern relating stomatal conductance to vapor pressure deficit to the minus 1/2. Now, if you read, again, John Norman's book, you find that there are several empirical relations that link stomatal conductance to photosynthesis divided by CO2. John actually worked with the Berry formulation, which is more of a relative humidity measure. Rayleigh Yuning in 1995 came up with a revised version that considered vapor pressure deficit, but the upshot of it is that these equations empirically suggest that stomatal conductance is linear in photosynthesis divided by atmospheric CO2. This quantity here is an emergent property of the optimization. It was not a priori assumed. The aridity measure that emerged is an optimality principle emergence, not assumed a priori. Now, I should say that these two models, by the way, still populate the climate models and the Earth system models that are being used today. So one more check on that is done by my colleague, Sari Pagroth, who measured photosynthesis and water vapor fluxes on Scots pine across a climatic gradient. And she plotted the transpiration rate on the y-axis against the photosynthetic rate adjusted by the square root of vapor pressure deficit on the x-axis. And across a whole range of climatic gradients for the same species, Scots pine, there was a complete collapse of the data on the same line, consistent with the prediction that the transpiration, which is stomatal conductance multiplied by vapor pressure deficit, scales as square root of vapor pressure deficit and is linear in photosynthesis. So that's an interesting observation that, of course, ended up corroborating these types of optimality solutions. And this somewhat result kind of brings to focus earlier observations by Francis Darwin, who we will revisit a little bit later. Francis Darwin actually was among the first to conduct experiments on stomatal closure due to what he called the withering and dry air. And he basically stated, well, by the way, Francis was fond of the hydroscope, yes, so he was able to measure vapor pressure deficit, and by measuring vapor pressure deficit and looking at stomata, he was finding that stomates actually shut down under high vapor pressure deficit. This was purely observational, and he noted that when we remember the endurable adaptations which serves to economize water, it is inconceivable that the closure of the stomata should not cooperate. There's a lot of not in this, but the Brits like that. you need to hold a lot of information to understand what they're saying. So in any event, the fact that stomatal conductance is related to d to the -1/2 is saying the same thing. As you increase vapor pressure deficit, stomatal conductance shuts down, but it shuts down in a very universal way, d to the -1/2. Okay, so this whole analysis was based on what we call static optimization, and they can be criticized, I would say, ad infinitum. But there are also ways to handle the variations in the marginal water use efficiency. And those tend to be labeled as dynamic optimality principles, and they're again using the calculus of variations, nothing new. Except that this time, the evolution of the marginal water use efficiency in time depends on not just the state variables themselves, but it depends on the amount of water in the routing zone. And this has been done by Asad Bragg, and it has been done by Saverio Perry. Especially in the case of Saverio Perry, he did that for salinity stresses rather than moisture stresses, a different type of salinity stress. Same thing, it has a similar impact as droughts. And Asad did that and showed in a minute, as we shall see, that you could recover a whole slew of strategies that have been reported in the literature, again, by using dynamic optimality principles. And the way he did that is that he reformulated the Hamiltonian as before, depending on the state and the soil moisture variables and the rate of change of the soil moisture variable, the Lagrange multiplier that is evolving in time. And then there is also a terminal correction. So what happens to the water at the end of the integration period now has to be factored in. And if you envision that if you link this terminal value at the end of the integration period to what you are trying to maximize, then if you relate it to the stored water in the routing zone, you could prescribe a priori a conservative water use strategy, an aggressive water use strategy, by dialing up this parameter or dialing it down. In other words, if you want a conservative water use strategy for the plant and you want to study what should stomates do as a trajectory in time, what you could then do is make that quantity very big, which simply says that if there is a lot of water in the routing zone at the end of this integration period, it has a lot of carbon value, basically. So it has a disproportionate impact on the carbon economy. And you can do the same thing if you want to prescribe an aggressive water use strategy. In other words, the aggressive water use strategy will simply say, don't store water in the routing zone, go ahead, try to consume as much as you can. There is no value in you storing water in the routing zone at the end of the integration period because it might be outcompeted by your neighboring competitors, for example. And so you could do that, you could combine that with the dynamic optimality principle, and you could show that with these types of optimizations, you could now formally recover what an aggressive water use strategy or what a conservative water use strategy does to the relation between stomatal conductance and leaf water potential, and therefore recover the whole spectrum of isohydrate to anisohydrate properties, strictly starting from optimality principles and prescribing a water use. Before, it was assumed that, for example, isohydrate or anisohydrate plants have an associated water use strategy by assumption. Now you could prove it mathematically through the Lagrange multipliers adjusted by a terminal leaf. Okay, that's all I'm going to say about the leaves. I'm going to move on to now the hydraulic transport, the water delivery system. And again, just a brief look at the history of that. Water is again moving from the soil to the leaf in the xylem. And there was a big question about the driving force. So to go back to Aristotle, there was a lot of observations that water was moving up in the xylem, but again, the driving force for it was not clear. And that triggered quite a bit of a heated debate between two camps at the time in the 1800s. One camp is labeled as the vitalists, and the other camp was labeled as the physicists. And in this debate, the vitalists, in summary, argued that living cells should play a major role in the transport mechanism. Now, I should say that they did not identify these cells. What are they? How are they moving water? Are they squeezing water up as you go up the tree? Or did they suggest the detailed transport mechanism associated with it? The physicists, on the other hand, argued that only dead cells are involved, and hence the mechanism of water transport in the xylem is completely dependent on physical properties, nothing else. And this debate raged until Edward Strasburger, who was a professor at the University of Bonn, did a wonderful experiment in 1891. And what he did is he put basically a living system in copper sulfate, and that basically is nothing but poison for living cells. And he found that water kept moving after all the living cells died. So clearly water movement in the xylem cannot be associated with the vitalist view. But he did not offer a mechanism or identify the forces that caused the water to go up from saturated conditions here to the least. So this was kind of an important experiment because in fact, Edward Strasburger already met the leaders of cohesion tension theory later on, Julian Nixon, and they met him a few years ago. They were inspired by these experiments, and in fact, these experiments reinforced their own view about the potential for using cohesion tension as an explanatory driver for how water moves in the plant. And again, they proved that by wonderful experiments. They actually showed that water can still move from saturated liquid water here all the way up to the leaf, despite the fact that there is positive pressure imposed on the leaf. And that was a major finding. It was clear at that point that water is not, rather than being pushed out, it's still moving up, despite the fact that the atmosphere around it is subjected to positive pressure. And this led to the so-called cohesion tension theory, that water is moving because of cohesion and tension all the way up to the leaf. Now, Francis Darwin, to go back to the same guy, he argued that believing in the ability of water to bear tension is to some of us equivalent to believing in ropes of science. And as happens in academia many, many times, there was some literature at the time of Darwin, especially the experiments of Berthelot and Jager, who showed that water can withstand high tensional forces. In fact, the original measurements suggested that water can be subjected to tensile forces up to 50 megapascals and still withstand. This was completely unbearable to Darwin because of the confusion between tension and shear. So Darwin's interpretation of tension was basically that of a shear, and it is well known that fluids, by their own definition, cannot withstand shear. In fact, if you are into solid mechanics, yeah, you relate stress to strain. If you are in fluid mechanics, you relate stress to rate of strain. And you get the same result, basically, the same governing equations. The only thing that will change is just the constitutive law. One is stress strength, you get solid mechanics. Stress rate of change, you get fluid mechanics. They do not change. And the fact that fluids can withhold tension but not shear was just not well understood. Now, there is some debate about the validity of these experiments. It was argued before, since the inception of these experiments, that perhaps what Berthelot and Jager measured was not cohesion, but adhesive forces of the water molecules on the cells of the tubes that they did these experiments in. But that was not true. There was enough evidence to suggest that that alone could not have explained the strength, and it was the really cohesive forces that have allowed water to withstand high tension. One last wrinkle was coming from the work of Haberland in 1914. This is Haberland the dad, not Haberland the son, who is considered to be the godfather of the birth control theory. So way more famous than the dad. But the Haberland the dad in 1914, who he was an Austrian scientist, very interested in water movement in the plant, and observed that vessels and tracheids normally contain both air and water. Already this is a red flag for cohesion tension theory. How could you have air when you're completely relying on continuous columns being pulled up by one single water molecule? And later on, John Milburn, again, wonderful experiments using acoustics, with a wonderful title, Listen to the Trees, basically put high-resolution microphones and was able to listen to the cavitation that was happening in the trees. Clearly demonstrating that cavitation, or the formation of air bubbles in the vessels of the xylem, is not an extreme phenomena. It was a natural occurrence. It was happening all the time. And that raised some questions, once again, about cohesion tension theory. And so to resolve it, we have to do a little bit more work to link xylem properties to the physics that is driving water to move. And of course, the understanding is now getting some urgency given the fact that there is clear connection between droughts and forest mortality. I don't think I need to explain why this topic is important, just other than to say, yes, droughts are observed at many parts around the world, long-term stations, and yes, they do cause mortality. And one of the ways to progress on this understanding is to try to understand what's happening in terms of how plants resist droughts and basically how they can handle cavitation in their xylem. So to do that, what we're going to do is try to understand basically a lot of the data sets that have been collected about the relation between stress and conductance. And this is coming from the so-called vulnerability curve, or percent loss in xylem conductivity. Often you might see pictures like this. This is taken from John Sperry, again, a leading figure in this field, where if you plot xylem tension on the x-axis and percent loss of conductance on the y-axis, you see that different species have very different responses. So in the case of riparian trees, these are trees that are accustomed to having a lot of water. You could handle tensions of up to 1 megapascals, and then there is a very rapid loss in xylem conductance. If you look at desert shrubs, the story is very different. Now you could handle stresses of up to 6 megapascals without losing much conductance. Pretty high tensions. So the question that we see to determine then is what determines if a species is resistant to drought, which is equivalent to saying what is the shape of this vulnerability curve. And the way we're going to frame this problem is pretty much inspired by soil physics. Again, just to make a pitch to John Norman's book, he has a nice chapter on that, and John Norman himself is trained as a soil physicist. And basically what these calculations would suggest in a minute is that there is a connection between wood anatomical traits and the shape of this vulnerability curve. But basically the ideas that are going to be presented first come from soil physics. And just let me say what the holy grail is in soil physics. The holy grail is this: if you know the particle size distribution of a soil, that enables you to estimate the pore size distribution of a soil. If you know that, can you predict all the hydraulic, thermal, gaseous phase diffusion properties, electric properties of a soil? Can you do that? And the answer is a qualified yes. And this qualified yes comes from percolation theory. Initially, I was naive enough to think that this might work also for plants, and I was proven wrong. It doesn't, and I will say in a minute why. But first, just the connection between the two. This is the percent loss in conductivity as a function of xylem pressure for ring porous and diffuse porous species. Ring porous, you could see the rings at the beginning of the growing season. As the growing season progresses, you see the pores are getting smaller. This is the wood anatomy of it, if you will. Next year repeats itself. If you look at the vulnerability curve, it looks pretty much like an asymmetric Weibull-shaped curve. On the other hand, if you look at another diffuse porous species, you find that at the beginning of the year, you progress during the growing season. The pores statistically are not different from the beginning and the end of the growing season, and again, it repeats itself. So basically, we know that perhaps a species like this is very good at transporting water efficiently, but is very poor at providing safety in terms of cavitation potential, whereas in this case, you are pretty safe in terms of not experiencing very rapid cavitation at the beginning, but your efficiency is perhaps much less. So to solve the shape problem, one needs to solve a whole slew of scales. I would simply mention a few, that if you start at the composition of the xylem vessels themselves, you find that these vessels are composed of either vessel members or tracheids. They are connected by membranes. These membranes have a pit field where you have a lot of pores. These pores are at the nanoscale. This is on the order of centimeters to tens of centimeters. This is on the order of micrometers. And of course, you are dealing with pathways that are going to go to tens of meters. So if you really want to do this brute force, you will have to resolve scales from nanometers to tens of meters in a very complicated geometry, which cannot be done in a brute force way yet. So what is the alternative? Well, the alternative is to try to see whether this problem can be reduced a little. I should say that this design is quite efficient also because cavitation and cavitation spread in the xylem can be viewed as a problem that involves disease spread. So you could think of, for example, a xylem that is infected with air and then an intact xylem, or resembling a susceptible population. And basically the spread of air bubbles is very much like the spread of infectious diseases in a susceptible population. In fact, the math is very similar, just to say. And maybe there are a couple of lessons to learn about containment of disease spread that can be learned from xylem designs, but that is for another topic. For now, we will distinguish two types of pathways for water. One is axial pathway, so water is moving in these big tubes. And the dominant mechanism that is dissipating energy is wall friction, basically. And therefore, there is a relation between the flow rate, the pressure drop, the radius, and the length of that tube. And that's easy, and if you integrate many of those, you get still, across all these tubes, you still get a version of Darcy's law. No problem there. The problem is that water also has to go laterally through those membranes. And this lateral flow is driven by different physics. It's in fact driven by the so-called Sampson flow. Sampson flow occurs when you have a thin sheet with a hole, and you're trying to figure out the relationship between the flow rate, the pressure drop, the radius of the hole, and the viscosity of the fluid. And it turns out that the flow rate is proportional to the cube of the radius. Whereas, for example, in this case, the flow rate is related to the fourth power of the diameter of the pipe, or the radius of the pipe. So very different physics. Okay, so this is for water flow. What about air spread? Well, this is kind of the basic idea of air seeding. Once air enters into the xylem system through cracks, through, I don't know, some beaver digging, some woodpecker smacking the wood, air is able to enter, and therefore it creates an embolized vessel. And basically, at the beginning, you might have some mechanical equilibrium between the air and the water here, between a functional vessel and an embolized vessel. This is the membrane separating the two. As the tension increases, you start seeing a deflection of this membrane, and as the deflection of this membrane increases, the separation between the membrane elements or the pit fields increases, and therefore air can penetrate into the highly negative water. And once air penetrates here, it will immediately expand and push the water out. So with this model, we started to try to see if we can link the anatomical traits to percent loss in xylem conductance using a very primitive network that is two-dimensional. So it would represent something like a radial vessel arrangements of a maple tree. And what we did is we said, okay, well, take the vessel lengths, take the vessel diameters. The longer vessels should have more connections to other vessels, so that's conserved, statistically speaking, from measurements. This is all anatomical. And then what you could do is inject air at random in different clusters, and then start to apply positive pressure, so you pressurize the air instead of creating tension, and then see what happens. And you allow air to jump from an embolized vessel to a water-filled vessel in one of two ways, either by expanding the pore size or by linking that pore size to the membrane thickness. You choose. The results are qualitatively the same. And if you do this calculation and you start increasing the air pressure, well, no surprise, you're going to start getting cavitation spread, and those are shown in blue with 2.5 megapascals. Isolated vessels also emerge, and that's an important point for xylem refilling, but we're not going to discuss that today. But basically, there are lots of segments of this network that become hydraulically isolated, but they're still filled with water. Okay, so that's important too for the recovery. And then you still have some functional vessels there. And that's a network model, very primitive network model. Let's see how it performs against a vulnerability curve. Again, the y-axis is the percent loss in conductance. The x-axis is the applied air pressure, which is equivalent to tension. And the measurements are shown in gray, whereas the model calculations from many, many stochastic simulations with different air seeding is shown in symbols in red. And while the agreement is not perfect, the fact that this vulnerability curve is reproduced strictly from anatomical traits is suggestive that this model is capturing the essential features of the vulnerability curve. Okay, as a bonus, as I mentioned, you could predict also the amount of water that is retained in this network, and that is often quantified by the relative conduit water content, and that is shown actually here. The y-axis is either the percent loss in conductance or the relative conduit water content, and the x-axis is the air pressure that has been used to basically increase cavitation. And you can see while the xylem vessels have lost almost all their conductivity by about 2.5 megapascals, at 2.5 megapascals you still have 50% of the water still in that network, which is pretty impressive. And that is going to be used quite effectively to basically alleviate the cavitation in the xylem system and refill the vessel. So there's plenty of water for that. And this is becoming also an interesting point to ponder more because of recent advances in remote sensing of plant water content from space. Alex Connings, former student who did a master's with me, she basically is now one of the leading figures in terms of trying to use microwave remote sensing for estimating plant water storage. with the premise that there may be a link between hydraulics and plant water storage. So this is an exciting area that is within development remote sensing. I was discouraging Alex from going to the remote sensing because I told her this is a saturated field, and again, she proved me wrong that she's doing great work in that field. So, last, just 2 minutes more, we're going to end with the flow. And I wanted to show that the same type of optimality principles that we have discussed in the xylem, which is kind of a safety efficiency with the leaf, also apply to the third transport system, which is mass flux in the flow. A lot of the early theories are attributed to Ernst Mach. And basically what you could see as an optimality principle is somehow sketched already. The sugar mass flux in the flow, J, is proportional to the water flow rate multiplied by the density of water multiplied by sugar concentration. So this is basically ignoring diffusive transport. But in this case, it's okay to ignore the diffusion one. The sugar molecules are sufficiently big, and therefore diffusion does not contribute as much as advection. Now, in low Reynolds number flow, you could still link the flow rate to the pressure drop and the viscosity of the water. This factor here is purely driven by geometric considerations, nothing else. There's no dynamics, it's only geometric. And the viscosity of the water, of course, varies with concentration. I mean, sugar maple, for example, is quite viscous because there is a lot of sugars in the water. And then you could formulate a model for the pressure differential. It could be hydrostatic, and it could be osmotic, or some of both. Osmotic here meaning that the increase in sugar concentration increases the osmotic potential. You take that expression for delta P, you plug it here, you take that expression for the flow rate with the plugged expression for delta P and the viscosity, you put it back in here, and you find that the flux will see two competing forces. If you increase the concentration, you increase the mass transport, but you also increase the viscosity. So there is some sort of a conflict. And sure enough, if you plot that, sugar concentration by weight, relative to weight on the x-axis against the mass flux on the y-axis, you find that for the majority of different assumptions about the pressure, so constant, osmotic, and the sum of the two, you will get some sort of a generic optimal. And by the way, that optimal that is predicted from this highly simplified model is commensurate to the range that is reported on average across very many species for optimal sugar concentrations or operating sugar concentrations. The lower values are for passive loaders, the upper values are for active loaders. There's a little bit of a difference between the model calculations and the optimal peak, but still, this is a pretty impressive conclusion given how simplistic this model is. And I should say that this difference is not trivial, and there is a lot of hidden physics behind it, which I will not. So now to conclude, which I'm coming to the end of, there is a whole new field in physics that is labeled as networks on networks. And since we have shown that there are different optimality principles operating underneath the xylem and the flow, certainly one might ask, is there a more general optimality theory that welds these parts together? And this general optimality theory up to today remains elusive, and it may not exist. However, there are some blueprints of how to formulate it, at least mathematically, that is coming from physics. Eugene Stanley from Boston University is one of the leading figures in that, and these are based on networks on networks. In other words, if you have a network and you have another network and you stitch them together, say a power network and a computer network, let's say, you stitch them together and one is operating the other, you don't have a lot of resilience in that network. When the critical node fails, the whole combined networks will fail. On the other hand, networks that co-evolve with other networks, so there is an evolutionary process where the network grows with the constraints from the other network, and therefore their co-evolution actually generates a network on network. These tend to be way more robust, and these are general results that are coming from network theory. We have also shown that it's possible to predict the shape of the vulnerability curve and the relative conduit water content from wood anatomical traits. This is exciting because now, for the first time, we can measure at high resolution all the scales basically in the xylem. Xylem vessel length, vessel width, even the pit diameter can be mapped at the nanometer scales. And now how to connect this information to vulnerability curve is an active research topic where some connections between fluid mechanics, microfluidics, and xylem physics is warranted. And again, this is kind of just repeating the same thing, that these multiple scales can be observed from X-ray tomography, MRI, and so forth. So this is an exciting area in plant hydraulics. Measurements are becoming available in ways that we have not had before. We are able to see certain things that we've never had before. And it seems that now data is ahead of theory. Yeah? The experiments are progressing. The ability to observe anatomical traits and the geometry of the xylem network is well ahead of the theories that look at the aggregate behavior. So with that, I'll stop. Thank you.
Questions?
Yeah.
So I'm intrigued by predicting vulnerability curves from anatomy. I mean, I've seen your work on that, but I'm wondering what you think about predicting the vulnerability curves of things that are really, I mean, stems aren't that hard to measure vulnerability curves on. So what about things that really are, like fine roots and leaves and should the principles should still hold?
I think the idea. So what makes this somewhat interesting? First, on the water side. From the water side, as long as you have wall friction on one end and you have Sampson flow on the other end. I mean, this is the physics that is driving water. So in this case, really, there's nothing complicated about describing the physics as long as you know the length and the diameters, right? In this one, it's a little bit more complicated, but not that different if you know the pore size distribution, right, of these pit fields. In fact, you could think of this pit field as an interesting case where, because the biggest pore tend to be the critical path, if we want to still use percolation theory, is the critical path at which air will escape from the embolized vessel to the intact vessel. You are almost using extreme value theory to dictate the shape of the vulnerability curve. And extreme value theory says, you know, if you repeat an experiment, you pick an extreme. You repeat the experiment, you pick the extreme. You repeat the experiment, you pick the extreme, or the highest value. What's the distribution of all these extremas? Well, it turns out to be an extreme value distribution. And that has a Weibull, Gumbel, or Frechet, or we look at a generalized extreme value distribution. So the connection between the shape and the pore distribution, the kernel of that integral is there. So as long as this is the case in the vessels of roots and in the vessels of the xylem and the vessels of the branches, I'm not that good at all, so I wouldn't be able to provide you with a very meaningful answer. But as long as the setup is the same, and as long as you, but more important for this network is getting the connections right. So as long as you know that for long vessels, how many connections on average there are, and this is the critical part, then you could do a lot, I think. And again, I mean, how you see this network has some effect, but those you could average over in the simulations, and those are never known. You know? And again, I mean, even the data from Frederick Lenz, I mean, this is the same species repeating the vulnerability curve, so you could see that there's quite a bit of spread also in the repeatability of the experiment.
Yes.
And probably just exactly for reasons of how, when you start ramping up the air pressure, let's say, where is that first bubbles beginning to form and expand. And those tend to have a huge impact on the initial conditions. I mean, the model simulations, by the way, have about, I didn't show them, but they have about the same bars, if you will, as those measurements, if not even more. But these are on some average of both. So this is my answer. When it comes to leaves, it's a little bit of a different network. I'm aware of that. Leaf venation is a big topic.
Sure. Well, and even outside the xylem. But
Yeah.
in a fine root, it seems like it might work.
Yeah, and so I think these are the ingredients of the model. And this is 2D. If you add a third dimension, it becomes a little bit more complicated. And this has been done by us. The code is actually on GitHub if you want to play with it, for both the 2D version and the 3D version. And it's basically still using air pressure as a surrogate for suction. But the idea is similar. Yeah, it's a guess. Again, exactly like soil physics.
There's a question from online. Raj Gopal asks, did you look at the variability of entrance region and root water flow that may affect the plant hydraulics?
Well, thank you. That's a great question. In fact, the answer for these specific calculations, no. These calculations assume that you inject basically air bubbles in this network at random. So there was no rules per se. It was just simply saying that if the network topology in terms of length, diameter, and connections is preserved, statistically, not mechanistically, you get that answer. But of course, if you're trying to model water moving from the soil all the way to the leaf, you have to look at the rhizospeed, you have to look at the root distributions. And this has been done. I mean, this is not new. The challenge is how do you generate vulnerability curves theoretically if you know all the anatomical traits? And it's almost asking the same question in soil physics. If you know the grain size distribution, can you predict the hydraulic properties of soils? I mean, that's almost the analog of this problem.
Thank you for a fascinating talk. Excuse my ignorance, but I just wanted to ask whether there's any communication or coupling between the pressure gradients in the xylem and the plant, or whether they're totally different.
There is certainly, and it was kind of not explicit here because, let me go to the equations. Basically, it is somewhat hidden here, right? So this pressure gradient that you have to work against is certainly, part of it is hydrostatic, but again, part of it can be imposed by the xylem. So in other words, when you inject sugar in the phloem, your osmosis has to compete with the negative pressure in the xylem to pull water from the xylem to the phloem. So that has to be high enough to be able to do that. However, the hydrostatic distribution, for example, may not matter too much because you're looking at pressure differential between the xylem and the phloem. But the concentrations that you need have to overcome the negative pressure in the xylem. And in fact, phloem failure is often attributed to the fact that if the concentration is not high enough, you actually get water moving from the phloem to the xylem, and that's it. I mean, if you think about the volumes, the phloem is a tiny fraction of the xylem, so you immediately empty the phloem, and that's it. You have a phloem failure. And that means that the plant is, for all practical purposes, dead. And the phloem is much harder to repair than the xylem.
A great talk, Gabby. Thank you very much. But my question is, what do you think about the limitation of the optimality theory? Are there any circumstances that that kind of theory is not applicable in the field of hydrology?
Yes, I can spend the whole day on that.
Yeah,
I don't think there's enough words to describe the limitations. There's plenty.
There seems kind of debate on that kind of theory.
Sure. No, I'm aware of the debate. And what I have proposed is not unique. There are many other objective functions, even though they take on similar form. Some gain minus some cost. Sometimes it is formulated as a relative gain minus a relative cost. So all of them have a similar flavor. What is advantageous is that they are somewhat broad statements that should apply to all plants. They are not particular to a single plant or a single species. And from that broad statement, you are able to deduce, very much like Maupertuis did, the laws of mechanics from an optimality principle. If you think about the principle of least action, you could derive Newton's laws from the principle of least actions. And we're kind of trying to come up with something similar, to not with the same success. But that's the goal, is that can we then find an operating principle that does not require information about all the complexity that you have in the stones, in terms of signaling, in terms of how the aperture opens and closes, what are the mechanical forces, what the adjacent cells are doing. So if you want to avoid all of that and go back to a formulation that is, so to speak, sufficiently minimal in number of parameters, that would be one possibility. If you want to also understand strategies, this would be a reference state. So even if the model itself is a failure, the failure tells you something that perhaps there is another limitation that you have not considered in your optimality rule. So it's a good reference state to compare. And even if it fails, it's informative. Because it highlights other limitations.
Thanks. Yeah.
We have time for one more question if there is any. Okay, if not, let's give our speaker Gabi a hand. Always I have to think about your work several times and then I get convinced that everything I know about this is wrong. And then you usually have something that makes me think again. Paul and Gabi, and I believe a couple of us may be heading over to the set afterwards for cold drinks. And so anyone is welcome to join if they want to continue the conversation. We can look at bubbles and beer instead and do some other sorts of physics. And then for the rest of the CPEP seminar series, there is not a seminar next week. But the week after that, we'll be switching gears back to thinking about policy and science. We'll be having Chelsea Chandler, who is the director of Clean Wisconsin, give a talk, and she'll be talking about science and policy. And then you'll see on the board outside, there's a few more talks, and that's going to wrap up our semester. So 3 more talks left.